So hello guys, welcome back. In this video we will study about the blood groups, that is the ABO system, that how a baby or how a child receives his or her blood groups. Let me first tell you that there are four main types of blood grouping system: A, B, AB and O. Later on, how it becomes AB positive or AB negative or B positive, B negative, AB positive, AB negative, or O positive or O negative. We will discuss it when we will study about the RH grouping system, which is the rhesus monkey. We all know that maximum times we get confused that how it is possible that a mother is A positive and father is B positive and the child is AB positive or AB negative or O positive, O negative. So here in this video today I'll discuss with you all about how it is possible or how a child acquires his or her blood. So talking about the blood groups, we all know that human being consists of red blood cells which are known as the erythrocytes, which are mainly enucleate, which means they do not have the nucleus, which is called as the director of the cell. But it may be noted that during the development or during the formation of erythroblast into erythrocyte, there are at least 4 to 5 stages through which the erythroblast goes in order to get mature erythrocyte. So the erythroblast have the nucleus, or we can say up to the end of a stage known as a reticulocyte. The red blood cell is having the nucleus, later on which it goes out by the process. Now we should first consider that suppose this is a red blood cell, or it is not biconcave in shape, but I am only making it in order to make you all understand. This is red blood cell. It's having the outer membrane which is mainly made up of the carbohydrates known as the oligosaccharides. Don't get yourself confused because different types of cell have different types of compositions in their plasma membrane or the outer membranes. Some are made up of proteins, just like in RH blood grouping system we will study that the outer layer is made up of proteins. So in order to proceed in this topic, let me tell you in detail. We will study the 4 different person having different type of blood groups. Person number 1, person number 2, person 3 and 4. Let us suppose all these 4 person or individuals are in the mother's womb or are children. Okay, so now we can see that, let's suppose this is a red blood cell which consisted of a nucleus. Only the mature erythrocyte do not consist of the nucleus, but the previous stages consist of a small nucleus within them. As we all know that inside the nucleus there are chromosomes on which there are different types of, or you can say that specific genes which are present in them. Gene mainly controls the hereditary pattern. There are millions of genes, or you can say uncountable number of genes inside the nucleus. Here I am only focusing about a gene number, sorry, not gene but chromosome number 9. So we were talking about chromosome number 19. This chromosome number 19 expresses a gene which makes an enzyme called as the fucotransferase. Remember, it is fucotransferase, not fucose transferase. So suppose this gene made an enzyme called as the fucotransferase, happy transferase. I already told you that red blood cells have an outermost membrane or outermost layer which is made up of an oligosaccharide precursor molecule. Suppose this is oligosaccharide precursor molecule, oligosaccharide precursor molecule, or you can say OPM. This person number 4, we are talking about person number 4. Later on I will tell you that how you can determine that what is his blood group or what was his blood group. This oligosaccharide precursor molecule, this happy transferase goes here. Suppose this is the shape of happy transferase. This happy transferase gets attached here with the oligosaccharide precursor molecule. This whole structure is called as H substance or H antigen. H substance or H antigen means no antigen. So we all know the blood group in which there is no any antigen is blood group O. I hope you understood this. Now coming on to person number 1, same case, erythrocyte or the coming on to the person 1, this is the erythrocyte or the previous stages of the erythrocyte, mature erythrocyte. This is the nucleus. In the same way, here are the chromosomes. In this either maternal or the paternal chromosome, this is chromosome number 9. They produces a substance or an enzyme, or you can say transferase, known as NAGA, an acetyl glucosamine. Okay, so this produces NAGA. This was OPM. This was attached fucose. So whole this was H substance. When this NAGA, you can shape it like this triangle. When this NAGA goes and attaches to the H substance, this structure is antigen A, and person 1 is having blood group A. So in other words, we can say that H substance plus NAGA equals A antigen. Similar case comes with person number 2. Red blood cell, nucleus, chromosome. Now what? Chromosome number 9. Allele. Gene is having an allele. Allele means variation of one gene. Means suppose there is, I am only giving an example, this is not a gene. Suppose there is a gene known as KH. It may have allele as KH1, KH2, KH3, or you can say there is a gene known as HK. It will have alleles as HK, HK2 or HK3. Similarly, here the allele of gene number 9 was producing Naga. Same gene number 9 here is producing galactose. Naga or transferase A, galactose or transferase B. Here it was, let me draw it, opium, this was fucose. Suppose star is a galactose. This was H substance, star adding to the H substance, resulting in the formation of B antigen. Person is having blood group B. Now the interesting person. Now you might have understood that what will be the blood group of the person number 3, it's AB. So what comes here? Red blood cell, the nucleus, parental chromosome, maternal chromosome, chromosome number 9. In this case, the parental chromosome may produce transferase A, maternal may produce transferase B, means paternal may produce Naga, maternal may produce galactose, or it can be vice versa, means parental can produce transferase B, maternal can produce transferase A. When both of these, triangle and the star, goes to OPM along with fucose, another OPM, this is star, this is triangle. Sorry for nuisance. This H substance, attachment of Naga plus attachment of galactose, antigen A and B, person is having blood group AB. I hope you all guys understood this. A little bit complicated information, but I hope you all understood this. Only thing was that you must remember erythrocyte have carbohydrate covered plasma membrane or the outer membrane or the cell membrane. Now talking about the antibodies. Antibodies are mainly produced by the B lymphocytes. Or plasma cells. All these things we will study in the immunity, when we will study about immunity. So what are the antibodies? Antigen is A, antibody will be B. Antigen is B, antibody will be A. Antigen is A and B. So what will be the antibody? No antibody. Antigen is no, antibody A and B. How the child immune system knows that whether to make antibodies A or B? So it all depends on the blood group. Person having blood group A have antigen A, so automatically antibody will be B. If it will make antibody A, then the antibodies will directly attack on antigen A and it will be hemolysis. So one question arises that whether the child is having the antibodies from the beginning. No. When the baby comes out, it receives the antibodies or the antigens entering into the body. Antigens are either the foreign substances which provokes